Yeah, it's kind of crazy, but yes, it's another good point. I gotta go.
Have to move. We have to move because we are very late on time, and we have the second talk by Gesine. Gesine.
I'll just take this one.
Okay,
Here, yeah.
that's great. Thank you.
Thank you.
Thank you very much.
Thank you.
So, for those who don't know me yet, my name is Gesine Heuck. I work with Precision Nanosystems, and if you were here earlier, you did hear a tiny bit about work that is done with our systems. And I would like to talk today about our nanoassembler platform that enables drug targeting and drug formulations from small scale development all the way to get to the clinic. And before I forget, I would like to make some publicity for this event. This is a symposium that we host in Seattle one day before the CRS conference. So those who attend the CRS conference, please come and join us. It's a free symposium. It promises to be interesting because we have a lot of interesting speakers like Yvonne Perry or Gaurav, who's an ex-Bob Langer MIT professor now at Oregon State University, and many more from industry and academia that present their work on drug delivery. So please come. If you cannot attend CRS, there is a live webcast, so also please feel free to sign. It's all for free. All right. So delivery is the key challenge to unlocking the potential of the genetic toolbox. I did steal this slide from my CEO. I'm not sure if this is some big person who he quoted or if this is his own quote, but I liked it, so that's why I took it or stole it from him, because I do believe it's true from my very unbiased opinion as a pharmaceutical scientist. The truth is also, though, that we still have a lot of bottlenecks and a lot of challenges that we face here, mainly having, limited capabilities. We have trouble controlling our nanomedicine compositions. All the methods that we still use are very bench scale focused, very labor intensive, very dependent on the actual person who performs the work. And therefore we have a lot of batch to batch variability. Scale up is a massive problem, and technology transfer is a massive problem as well. So what if we could change that and could make our lives much easier? Well, that is really the idea of Precision Nanosystems and of the nanoassembler platform that we have. So we have a number of solutions here, small instruments, where we start from reagents going over to low volume devices, larger bench scale devices, and then scale up. And even we have a GMP platform that enables you to develop your siRNA or mRNA oligonucleotide nanoparticles for gene delivery, and also other delivery devices that I'm happy to talk to you about at our booth. And the underlying method of the whole thing is really one core piece, which is a microfluidic technology. And so here I would really like to rush you through a few examples and maybe show you how these oligonucleotide nanoparticles are made, and just a few examples on who applies this and how they apply it. And so how do you actually make these RNA lipid nanoparticles in the microfluidics? I think Nidhi already mentioned this quite well, but we have our microfluidic cartridge, and really what we have is just a microfluidic channel with a microfluidic mixing chamber that features these specific staggered herringbone mixers. These staggered herringbone mixers allow us to mix two fluids and two intermiscible fluids in a very fast manner. This is within a couple of milliseconds, and it's in a very confined volume. So if we were to make siRNA or mRNA lipid nanoparticles, we would inject the RNA in an aqueous buffer on one side of the channel, and then lipids, different types of lipids, it's a cocktail of lipids into the other channel. And it just gets injected in there, and it gets mixed. And what comes out, if I have the right slide, oh, let me jump to this slide, we actually get to these very complex structures. Now, how does that work? And just to tell you what this is here, so this is a cryo-TEM image. We can see these are, very complex structures, and we can see that these have an electron-dense core. Now, how does this work? We inject the RNA on one side of the channel, as I just said, in the aqueous phase, and then the lipids, which are all these colored dots here, on the other side. And what happens is that this RNA, highly negatively charged, interacts first with a cationic lipid, and it gets completely complexed and forms these clusters that are now lipophilic. What happens then is that other helper lipids, this one here, for example, is cholesterol, or these purple ones, this is another helper lipid, they assemble around these RNA clusters. And last, we have these amphiphilic, in orange, these amphiphilic pegylated lipids that assemble around the whole lipophilic cluster, and then form these nice round lipid nanoparticles that encapsulate and protect the RNA on the inside. So, as a first example, how does that actually apply in vivo and in vitro? The nice thing about these lipid nanoparticles is that at pH 7.4, physiological pH, they are neutral. And so this is, let me go back here, sorry. First, obviously, we work with cationic lipids, so these are positively charged. But when we are at pH 7, as I just said, they are neutral. That means they are really not toxic. They don't interact with negatively charged cell membranes. However, what happens then is that they get internalized into the cell. And this actually happens through an endogenous mechanism. It's not passive endocytosis. It works with through an LDL receptor mediated mechanism, or depending on what some other users do, they just use antibody targeted delivery into the cell. What happens in the endosome? Well, the endosome becomes a lysosome, pH goes down, and then these particles, or these lipids, they get positively charged again. So now this positive charge can interact with a negative charge of the lysosome, and the RNA gets delivered into the cytosol and can do its work there. And this is just an example from an early work in 2013. This is from Brian McVicker's lab at UBC, where they tested these lipid nanoparticles on very difficult to transfect cells, which are neurons. And here, this is an in vitro experiment. This is just the cell marker. But what we can see here in red is that these particles were very nicely taken up. And in fact, this was an ApoE-dependent mechanism. And then they injected this into the cortex of a mouse. And this time, the particles are marked in green, but that shows very nicely that there's distribution of the particles throughout the cortex. And this is the knockdown results from the same experiment. So in vitro, they were able to specifically and efficiently knock down a housekeeping gene, P10, for over 21 days. And when they injected into the cortex for a distance of up to 15 let me see if this was 15 days, they were able to knock down the P10 in vivo as well, quite efficiently. This is one of our very recent images. I like it because I think it looks like an artwork, where not only we were able to transfect siRNA, one type of RNA that's much longer and more difficult to transfect is mRNA. And here we successfully were able to express GFP in astrocytes, as we can see here. So in red we have the astrocyte marker. Here in light blue, this is the marker for the lipid nanoparticles. And this is here GFP, as we can see. And we get an effective transfection of the GFP mRNA and expression of the protein. These systems have by now already, I mean, the system is a few years old, well, 3 years old, and they have by now been used quite a bit in vivo, and these are the different injection sites that we have already seen and validated, or other people have validated. Going to the brain research project, what might be of interest is that there's a lot of work being done on on the brain and in neurons in general, but people have also used it for the liver, for blood diseases, and just 3 examples now that I want to rush you through, just to show you a bit what's being done. This work here is also at Brian McVicker's lab at UBC. He uses these particles a lot to study neuronal swelling. So this is what happens when you have brain injuries. There is a lot of influx of sodium and calcium ions into the cell, and by using these siRNA lipid nanoparticles, he's been able to study the function. So it's unclear what exactly the physiology is there and how this works. And just by using these lipid nanoparticles, he's been able to study the function of the physiology of this whole neuronal swelling process. This work is done by Dan Pear here. So Dan has he already gave a talk, which was on a different publication of his, but this was a proof of concept to target T cells. And what Dan does, it's quite interesting. So he makes these siRNA lipid nanoparticles on this microfluidic cartridge and then decorates them with antibodies to target these T cells. And what you see here is that when he targeted the CY5, CD4, decorated with CD4 antibody, and then targeted the CD4 receptor. And if he didn't target with the CD4, he didn't get any uptake into the cells, but he was able to specifically target T cells through the CD4 receptor with these lipid nanoparticles. He then used RNA, CD4 mRNA, CD4/5 mRNA to express that in the cells, and successfully was able to transfect the CD4/5 mRNA, the CD4 siRNA, 4/5 siRNA through the CD4 receptor, and was able to get a very efficient knockdown in these T cells. Also knowing that T cells are, like leukemic cells, not easy to transfect. So that was a pretty exciting finding. And this last paper that I just want to point out, so I put the references all in this presentation. This work is done at Pfizer, who focused on Friedreich's ataxia, which is a neurodegenerative disease, and they successfully transfected frataxine mRNA in the brain in vivo. Please come and see me if I can give you more details on this. I know it's very short, all of this. Just want to point out that work on DNA is also done. This was done at the University of Aston. Quite exciting. DNA being also a large molecule, very hard to actually put into particles and get to work on it. And I haven't spoken very much about clinics, but I do want to point out that all these particles and all these studies have been made with a small volume on the benchtop instrument that we have, but the platform foresees that when you have something where you can do a proof of concept that works in vivo, you can immediately use a larger instrument and make a larger volume with the same type of microfluidic technology, and we'll get the same results, quality results of these particles. All right, I'm done. Please, as I said, come and see it as the booth. We have a booth here. I'm more than happy to chat more about the technology, and I thank you for your attention.
I'm very sorry.
No, that's all right.